Last time I finished talking up about the citric acid and the glyoxylate cycle. And today I want to start bringing together metabolism to you in what I hope is a real fashion, where you can relate to it with respect to what's happening in your body and basically to physiology. This involves two systems that we need to consider: electron transport and oxidative phosphorylation. Probably two things that you have learned about in other classes like BB314 or even in general biology classes. It's in electron transport and oxidative phosphorylation where the oxidative energy of the citric acid cycle and of glycolysis are converted into ATP energy. And this is the major source of ATP that occurs in our bodies by a long ways. There's virtually nothing that compares with the ATP energy that's produced in electron transport and oxidative phosphorylation. Last time the time before last, I showed you this image of a cross-section of a mitochondrion. And in the mitochondrion, we saw that there was a matrix, and it was in the matrix where most of the reactions of the citric acid cycle occurred. In fact, all of them occurred if we included the enzyme that was embedded in the inner membrane. I also told you that the inner membrane of the mitochondrion is very critical. It's very, very impermeable to most substances. And that's particularly critical for us today because it is impermeable to protons. So protons cannot readily move across the membrane unless something is done to the membrane. And the only way to move protons across the membrane are either through a transport protein that we'll talk about. That transport protein makes ATP, or alternatively if the membrane is damaged, in which case the protons bypass the transport protein and leak into the inner mitochondrial membrane. Well, you might wonder why protons would want to pass through the membrane in the first place, and the answer to that question is because there's a gradient. There's a difference of protons on the outside of a mitochondrion compared to the inside. And this outside gradient, a higher concentration of protons on the outside compared to the inside, this higher concentration outside gets there as a result of action of the electron transport system. So the electron transport system is involved in creating a gradient of protons, and the oxidative phosphorylation system uses that gradient to make ATP. So in simple terms, that's two sentences that describe electron transport and oxidative phosphorylation. Okay. Now, where do we get the electrons that are used to drive electron transport? Well, you remember from glycolysis and from the citric acid cycle that electrons are stored in electron carriers in the form of NADH and also in the form of FADH2. Electrons from both of those complexes will enter the electron transport system, and as they move through the system, they pass from a series of molecules/proteins, both are involved actually, and we can follow their transport by looking at the energy of each of those transfers. And we see that overall there is a net downward movement in the direction of the electrons being transported. We can think of this almost like a voltage, because the electron transport system behaves very much like an electronic circuit. Very much like an electronic circuit. It's not instantaneous like a wired circuit is, but it's very, very efficient, and it moves electrons through it. I don't like that. This schematic shows the movement of electrons in the electron transport system. So let me set this up for you a little bit to where I hope you'll be able to understand this. What you see on the membrane here, the lipid bilayer, is the inner membrane of the mitochondrion. Now, you remember I said earlier that the outer membrane was fairly porous. A lot of things went through it. I described it as sort of a wet blanket. It didn't block everything, but it just kind of laid around the rest of the inner membrane. The inner membrane is where the mitochondrion really does its magic. Okay? In the inner membrane, we can see several things. We can see 3 large complexes that are involved in electron transport, and they have some fancy names. We're going to call them complex 1, complex 3, and complex 4. And you might say, well, where's complex 2? And complex 2 is not shown in this figure, but complex 2 behaves very much like complex 1, and I'll explain that in just a minute. Everybody with me so far? Okay. So between these complexes, we see that we have smaller molecules, in the case of this molecule right here called coenzyme Q, or small proteins, like we see here, called cytochrome C. So we can think of these smaller molecules as shuttling electrons between the larger complexes. The electrons come in the case of NADH, through complex 1. NADH donates its electrons to complex 1. Complex 1 donates those electrons to coenzyme Q, which in turn donates electrons to complex 3, which in turn donates electrons to cytochrome C, which in turn donates electrons to complex 4. And what does complex 4 do when it gets those electrons? It donates them to oxygen to make water. So what you've seen already is the movement of all of the electrons from NADH, starting over here on the left, and ending up over here making water. Okay? It's a series of passages of electrons, and we don't need to know all the details in here, the FMN, the FES, all that sort of thing. We're not concerned with. If you know that electrons move from NADH to complex 1, and from complex 1 to coenzyme Q, and from coenzyme Q to complex 3, and from complex 3 to cytochrome C, and from cytochrome C to complex 4, and finally to oxygen to make water, you know the electron transport system as far as I'm going to ask you in terms of how that transfer occurs. Okay? It's a fairly straightforward system. Now, believe me, it's very complicated. These complexes that you see here are called complexes because they're a large collection of proteins. They are large collections of proteins. And they have things in them, like you can see here, iron sulfur, that allow them to transport electrons through them. Now let's talk a little bit about the movement of the electrons themselves. The movement of electrons through complex 1, the electrons come in pairs. Okay? Electrons come in pairs. You remember that NADH had a pair of electrons that it had picked up from oxidation. Coenzyme Q has a very useful ability. It's what I call a traffic cop. Okay? Coenzyme Q, as we will see, also interacts with complex 2. But even more as a traffic cop, coenzyme Q is able to accept 2 electrons at a time, but pass them off to complex 3, 1 electron at a time. Now that's important because complex 3 and cytochrome C and complex 4 can only handle electrons 1 at a time. So thanks to coenzyme Q, electrons are passed through in the right fashion. Complex 3 is something we can think of as a docking station. In fact, complex 1 is a sort of a docking station as well, but it's more apparent with complex 3. Complex 3, on the one side, interacts with coenzyme Q, and on the other side interacts with cytochrome C. So the electrons are coming in from coenzyme Q, passing through complex 3, going to cytochrome C, and then cytochrome C goes over here. Each of the big complexes, as I said, serves as a dock. Here's the electrons, coenzyme Q docks. Let's go, goes over here and docks, passes electrons to the other thing docked, which passes electrons to cytochrome C, which goes over here and docks. So these smaller molecules are very mobile in the membrane. They're fairly easy to move back and forth, whereas the complexes themselves are not nearly so readily moved. Okay? You may remember we talked about, yes?
You said that it can accept 2 electrons, but only gives away 1 at a time.
Yes.
So what happens to the second one?
It waits until it's ready, the system's ready for the second one.
Okay.
So it passes them off one at a time. It'll pass the first one, the system takes it, then it'll pass the second one, and the system takes that. Okay? Okay. We've talked about heme. We talked about heme being involved in carrying oxygen. We'll see heme is involved in the cytochromes. These are cytochrome proteins that contain a heme group. So heme is also very important for electron transport. Most cytochromes contain at their center an iron. Complex 4, unlike the other complexes, has a heme that contains at its center, one of the hemes contains at its center copper. Now, how is it that electrons move through a heme, for example? Well, remember, first of all, that iron can exist as iron plus 3, that's the ferric state, and iron plus 2, that's the ferrous state. When the ferric state, plus 3, accepts an electron, it becomes the plus 2 state. And it can take that electron that it has accepted and pass it off to something else, where it becomes the plus 3 state again. So that we see by going between plus 3 and plus 2, iron is holding onto an electron and then passing it on to the next thing. Similarly, copper, which is over here, can exist in the plus 1 and the plus 2 state. In the plus 2 state, if it accepts an electron, it becomes a plus 1. When it donates that electron, it flips back to becoming plus 2. Okay. It's all making sense? It's a series of events. Bang, 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 bang. Now we're going to see one of the things that's that I'm going to show you after I talk about oxidative phosphorylation is that there are things that can block electron transport. And some of those things we know as very poisonous things. And I'll say something about those when I come back to them. Before I talk about that, though, I want to talk about complex 2. Because complex 2 is not shown on here. But complex 2 is also very important in the electron transport scheme. Complex 2 also interacts with coenzyme Q. And then once it interacts with coenzyme Q, it passes its electrons to coenzyme Q, and everything else, like you see on here, is exactly the same. So what is it about complex 2? Well, complex 2 accepts electrons from FADH2. That's the only difference for our purposes. It's accepting electrons from FADH2. Okay? So we have a system of electrons passing through, and we see over here this involvement of oxygen. The involvement of oxygen. The reason that you breathe is because of this guy right here. You've got to have oxygen in order for your electrons to have something to bind to. If you don't have oxygen, what do you suppose is going to happen? Well, this reaction isn't going to occur, right? If this reaction isn't going to occur, then this guy is full of electrons, which means it's not going to accept electrons from this guy, which means it's full of electrons, and we see the system backing up, backing up, backing up to here, right? Which means that NADH is going to have no place to donate its electrons, which means that FADH2 will have no place to donate its electrons. And if you can't have NADH or FADH2, what happens to the citric acid cycle? This is why you suffocate. If you don't have oxygen for a long enough period of time, you will suffocate because you can't run the citric acid cycle. Yes, sir.
For cells that produce the built-in oxygen for the final electron receptor,
Yes.
the same layout?
Yeah, that gets actually a lot more complicated. But there are a few cells that use things besides oxygen as the final electron acceptor. Many of them are much lower energy cells. They're all unicellular, and they will almost always rely much more on fermentation than this. You won't see this in a higher order organism. Yes, ma'am.
In the electron transport chain, does it use complex 1 or complex 2?
Well, it does use both because the cell has both NADH and FADH2.
Right, but one's for NADH and the other one's for FADH.
Correct. Correct. But remember, we have thousands of these going on in every mitochondrion that we have. So the NADH and FADH2, which are generated in the citric acid cycle, both enter. One enters through complex 1 here, the NADH, and the FADH2 enters through complex 2. Okay? Clear as mud. Okay. So that's what's happening with electron transport. If you really want to get complicated, there you go. We're not going to get complicated. Okay? Coenzyme Q, I told you, was the traffic cop. And no, I don't care if you know these structures. I'm just going to say a couple of general things about coenzyme Q. You can see from this diagram that it exists in a different structure than it exists here, than it exists from here. This is a 1-electron transfer. This is a 2-electron transfer. This is the reduced form, and this is the most oxidized form. Okay? So all the figure is showing you is what I told you is true, which is that it can accept electrons 2 at a time, becoming this, and let them off 1 at a time, becoming this. Okay? So it goes doink, and doink, and doink. That's what it's doing. All right. The other thing I'll show you about this is the R group. Look at the R group on coenzyme Q. The R group is a long chain of carbons and hydrogens. Is that polar or nonpolar? Nonpolar, you betcha. Where do you suppose that guy is going to be sitting? It's what anchors it to the lipid bilayer of the mitochondrial inner membrane. Okay? By the way, coenzyme Q, which is what I usually call it, or you can call it CoQ, is also known as ubiquinone, but you don't need to know that ubiquinone. Well, what matters for our purposes isn't just what happens in electron transport, but it's actually the combination of electron transport and oxidative phosphorylation. So in order to understand the bigger picture, we've got to start talking about the oxidative phosphorylation. I'm going to start right here. This figure is very much like the one I've been showing you. However, it's got a new wrinkle. It's got something I didn't tell you. And the thing that I didn't tell you is that as the electrons move through complex 1, 3, and 4, each time the electrons move through there, protons get pumped. They get moved from the matrix, which is on the bottom, to the intermembrane space, which is on the top. And the intermembrane space is between the inner membrane and the outer membrane. That's what the intermembrane space is. So the movement of electrons causes protons to be pumped. That means that the concentration of protons in the matrix will be lower than it is in the intermembrane space when this system is operating. Okay? Now that turns out to be critical because I told you earlier that we made ATP from the proton gradient. And this is how we make the gradient. So we make ATP from the proton gradient. If we can't make the gradient, we can't make ATP. Okay. So again, the same figure, things moving through. Don't worry about the complexity inside. But ultimately, donating those electrons to oxygen to form water. Well, the upshot, again, as I said, is that we get a gradient of protons that happens as a result of electron transport, And this is the other critical thing. Remember that this lipid bilayer is impermeable to protons, meaning they can't just come scooting back in. They can't just come scooting back in. They've got two options, as I said. Most of the time, the only option they have is to come back through a transport protein that's found in the inner membrane of the mitochondrion. That's this incredible protein called ATP synthase. ATP synthase. Okay? Now this is drawn upside down from the way before. Matrix is on top here, intermembrane space on the bottom. So you have to sort of reorient your head for that. Okay? But what is this guy? Well, let's look at it schematically. It's actually a series of proteins, as you can see. And this series of proteins looks something like this. Okay? It has something we call a stalk, F0, and that stalk is embedded in the lipid bilayer, as you can see, meaning there's a bunch of nonpolar stuff anchoring this guy in here. It's also got an F1 head, and it's in the F1 head where ATP is actually made. Now, I like to liken this guy to a water turbine that's used to generate electricity at a dam. In a dam, we have water higher on one level than we do on the other level. And so water to get from up here to down here has got to pass through a turbine. And passing through the turbine spins and electricity is generated. In this case, what we have is we have a higher water level, this case being a concentration of concentration of protons. Don't confuse protons and electrons like I just did. A higher concentration of protons in the intramembrane space here compared to the matrix here. Where do those protons want to flow? They want to flow from here to here, and the only way they can get through there is by passing through this chamber here. What does that do? Well, what that does is the movement of protons through this guy causes this little tiny protein that you see here in the middle to spin. It is a turbine. It causes this little protein, it's this guy right here, okay, the gamma, causes this little gamma to rotate. What does that mean? Well, because it rotates, as we will see in a second, there are three pairs of subunits called alpha and beta that surround this guy. They surround the gamma. It's in the beta subunits where the ATP is actually made. Okay, that wasn't what I wanted. All right. Imagine we're looking from above at the head of that mushroom. That is the ATP synthase. I like to call it the mushroom. Okay? Mushrooms have a stalk and they have a head, right? We're looking at the very top, from the top down. We have the alpha, beta, and we're only looking at the beta subunits here for each one. So we see three beta subunits. I said we had three pairs of alpha betas. So we have three beta subunits. And those three beta subunits are identical. They're absolutely identical. And you say, well, what the hell is the LTO then, Ahern? The three identical subunits change configuration depending upon which way the gamma subunit is pointing at them. So if the gamma subunit points in one way, it's an L. If it points in another way, it becomes a T. And it points in another way, it becomes an O. What's an L, a T, and an O? Slightly different configurations of the beta subunit. The L is what I call the loose configuration. It's able to bind to ADP and phosphate. That's the function of the L configuration. In the T subunit, in the T structure, the beta subunit is tightly squeezed together. The ADP and the phosphate are squeezed together, and that causes the formation of the ATP bond. So we've bound the ADP and the phosphate in the L. We've made it into ATP in the T. And in the O, O stands for open. What happens in the open state? ATP is released. So this guy flips between L, T, O. I don't show O down here. But you can see what's happening over here. Here is T going to O. T going to O here. That will next go to L. Here is O going down to L, and that'll next become T. So we can think of this as LTO, LTO. Every subunit is going through an LTO, and every time it goes through one full cycle of LTO, one ATP is made. This goes on rapidly. You can make thousands, or at least hundreds of these ATPs per second with this. It's really scooting. So let's refresh. We're looking at the beta subunits of the ATP synthase. The beta subunits are all identical, but their configuration differs depending upon where the gamma subunit is pointed. If we think about it, it's starting as the L. The L can bind ADP and phosphate. The T squeezes them together to make ATP. And the O opens and lets the ATP out. Questions about that? Yes? What makes it spin? That's a good question. What makes gamma spin? What's making gamma spin is the fact that protons are moving through that base. That's the driving force for the spin. Just like the water moving through the turbine at the dam, protons moving through that base are what's turning that. Okay? Yes?
To be clear, there are also alpha subunits that are not.
There are alpha subunits, and I'm not showing them here for clarity purposes. Yeah. There are 3 alpha subunits, one with each beta subunit. Okay. Let's look at one other thing, and then we're going to talk about respiration for the rest of the period. Look at this guy. This guy is a molecule called 2,4-dinitrophenol, or 2,4-DNP. Okay? This was a miracle diet drug about 100 years ago. You guys know these miracle diet drugs. You see them on the internet. You see them, God knows where, what magazines that are out there, right? And they all say the same thing. Melts away fat while you sleep, right? You take it before you go to bed, and you wake up in the morning, and all of a sudden you're this slim, trim Uber model or something. I don't know, right? Okay? The idea that you can lose weight while you sleep is a very big fantasy for a lot of people. Well, that's what this guy was advertised as 100 years ago. You could lose weight while you sleep. And it worked. And it does work. It kills about 10% of the people who try it. Okay? And we'll see why that happens. But yeah, people who took it, and they said, it's a miracle. It is a miracle drug. This thing, I take it in the morning, I wake up, I take it at night, I wake up in the morning, and I've lost a little bit of weight. How does it work? Well, what this thing does, they didn't know at the time, but now we know what it does, is it pokes holes in the inner mitochondrial membrane. Now let's imagine if you poke holes in the inner mitochondrial membrane, what happens? Let's imagine that we've got electron transport going. Let's imagine, first of all, we haven't poked any holes. We've got an intact inner mitochondrial membrane. Everybody with me? We've got an intact inner mitochondrial membrane. NADH comes along from the citric acid cycle. Okay? Dumps electrons into complex 1. Complex 1 passes it through. They ultimately get to oxygen, and water is produced. Proton gradient is made. Proton gradient is released by synthesis of ATP. Right? Makes sense. That's what I've been telling you all along. Electron transport followed by oxidative phosphorylation. What I didn't tell you was that ATP synthase requires ADP. Oh, it's not on here, but in the last figure, that ADP had to bind into the L configuration, because if it didn't, the whole thing stopped. ADP is necessary. And you say, well, that's fine. Okay, well, it's fine. Here's how your body normally handles it. I'm sure you guys all enjoyed the 3-day weekend we just had. I know I enjoyed it. And though I did a fair amount of work in the yard, I also had some time to sit on my butt and do nothing. When I'm sitting on my butt doing nothing, what happens to my ADP concentration? It goes up because I'm not burning ATP. I'm sorry, it goes down. My ATP concentration goes up. My ADP concentration goes down, right? I'm not burning ATP, and the only way I get ADP is if I burn ATP. My ATP concentration is high. My ADP concentration is low when I'm sitting, right? Everybody follow that? ADP stops. There's no ADP. What's going to happen to ATP synthase? Going to stop. If ATP synthase stops, what happens to the proton gradient? It grows to a point. But just like if I keep trying to push water, pump water uphill, the more water I pump up there, the harder it is to pump more, right? So that gradient gets so high that ultimately the protons stop being pumped. If protons stop being pumped, what happens to the electron transport system? It also stops. It's why we breathe less when we're sitting around. We don't need as much oxygen, right? Physiologically, that makes sense. I stop breathing because my body's not using oxygen. I don't need it, right? My electron transport system is stopping. What's happening to my citric acid cycle? If my NADH is increasing, right? What happens to my citric acid cycle? It's going to stop. And I know that if I don't exercise, I'm not going to lose weight. And how do I burn things off? Citric acid cycle. If I exercise, I can burn things off, right? So the more I sit around, the more likely I am to not do a lot of oxygen breathing. The more likely I am to be not burning off what I eat, because I'm still eating, right? And the more likely I am to gain some weight. Everybody with me? Now, I take the miracle diet drug, and I say, okay, well, I had a nice day, but I think a little bit of this 2,4-DNP will be great for going to bed tonight. I'm going to wake up in the morning a couple pounds thinner. I take my miracle diet drug. It pokes holes in my inner mitochondrial membrane. I doze off into sleep. Let's think about what happens if I survive it. Okay? What happens if I survive it? What happens? Holes are poked in the inner mitochondrial membrane. Okay? What happens when my ADP concentrations fall low? ATP synthase would slow down, right? But the problem is that my ATP concentrations don't rise. We'll see why that happens in a second. This is the key to the whole thing. Okay? When I poke holes in the inner mitochondrial membrane, I uncouple oxidative phosphorylation from electron transport. I uncouple them. They're no longer one dependent upon the other. As my electron transport system pumps out protons, and I'm not burning ATP, what would normally happen? Everything would stop, just as I described it to you, right? But what if I pump out protons, and the protons go out and they come right back in? What happens to the proton gradient? It's destroyed. And if the proton gradient is destroyed, what happens to my ability to make ATP? I can't make ATP. Does my body want to make ATP? Even though I'm lying down, you betcha it wants to make ATP because I have to keep things going even when I'm asleep. Right? So we see that there's nothing to slow down electron transport. Normally it'll slow down when that gradient gets high, but now there's no gradient whatsoever. And when the body sees no gradient, what does it do? It starts up electron transport. And how does it start electron transport? With citric acid cycle, making NADH and making FADH2. So the citric acid cycle starts like crazy. It makes NADH. NADH goes and dumps electrons and goes back and goes to the citric acid cycle again, keeping that going. Right? Electrons pass through the system, but no net protons go out. They go out, they come right back in. My system is burning energy like crazy, just like that miracle diet drug said it would do. Okay? But I'm making no ATP. That's why people died. Their system was burning and burning and burning and burning and burning, and no ATP was being made. They were uncoupled. The coupling means that you have to have an intact mitochondrial inner membrane. If you don't have that, they cannot be coupled. When you uncouple it by poking a hole in the inner mitochondrial membrane, okay, then electron transport is not stopped by oxidative phosphorylation, and oxidative phosphorylation won't occur because there's no proton gradient. That's nasty. The next question I always get, and I'll ask it before you do, is what if you took just a little? To which my answer is, what if you took just a little arsenic? Would you take just a little arsenic? I don't think you would. But there are people who will do this. Okay? Don't even dream of it. Okay? Yes?
So for those who survive, does the system ever repair itself, or is it permanently damaged?
So for those that survive, is the system permanently damaged? I'm not in a position to answer that question, to be honest with you. But my strong advice would be never mess with it. And there are people who are trying to revive this idea. Okay? Yes. Yes. So be careful. Be careful. It's not a marketable thing, but I'm sure we've seen the internet things that are out there about things that are already illegal that we can get. Question? Okay. Yes, back in the back. It's called 2,4-dinitrophenol or 2,4-DNP. Andrew.
Because there's holes, the whole electron transfer chain is still going.
Yep.
But the overall concentration of electrons is decreasing, right? Not the gradient, but
You're talking about the protons or the electrons? So the concentration of protons is equalizing between the two. That's what's happening. Instead of there being a gradient, you're getting outside and inside the same. Well, if the total number were increasing, what would you say was happening in the cell? Well, you would say that the buffer isn't working. Cells have to work in a very narrow range of pH, so that's not happening. Yeah. Let's think about what's happening. Go ahead.
so you have really rapid breathing during the night.
Okay, you're getting you're asking exactly what I'm headed towards. Okay, would you be breathing rapidly during the night? What do you think? You would, because you still are doing electron transport, and you still have to have a terminal electron acceptor. You'd be breathing like crazy.
It's like you're running, but you're sleeping.
You're running, but you're sleeping. And there's one other thing that would happen, and you guys should be able to predict it based on what I've told you about glycolysis in particular. You're going to get hot. You're going to get hot. Okay, those two things are going to happen. Right? You say you were hot in bed last night. I hope that you weren't talking about 2,4-DNP is all I can say, right? Okay, bad joke. All right, bad joke. All right. Now, I use this 2,4-DNP to start to introduce you to the idea, okay, of respiratory control. Respiratory control occurs when we have tight coupling between electron transport and oxidative phosphorylation. That's the normal state of affairs for our body. Let's go through a scenario. Let's imagine that I've already told you what happens when I'm sitting around doing nothing. I told you my ATP concentrations increase, right? Let's imagine I decide to go off on my morning jog. What's going to happen? Well, I take off my ATP concentrations. First thing that's going to happen is my ATP concentrations are going to go down. When my ATP concentrations go down, what happens to my ADP concentrations? They go up. When ADP concentration goes up, what happens to ATP synthase? Starts spinning. When it starts spinning, what happens to the concentration of protons outside versus inside? Do protons move or not move? Protons move. Okay. As protons move, what happens to the proton gradient? It's decreasing. When the proton gradient is decreasing, what happens to the electron transport system? It increases because there's less pressure holding against it, right? Electron transport increases. What happens to oxygen usage? It increases. That's why I start breathing heavily when I start jogging, right? I need more oxygen to put those electrons onto. As my oxygen use increases, what happens to my concentration of NAD? It increases because I'm dumping more electrons into the electron transport system. NAD concentration goes up. What happens to citric acid cycle? It increases. Now do you see why I exercise to lose weight? Makes sense, right? Okay. Now what we're going to see, I'll start talking next time about lipid metabolism. And one of the things that you're going to see is that oxidation of fatty acids involves production of acetyl-CoA. We get acetyl-CoA from oxidation of fatty acids. If I'm really good about my exercising, the best way to burn fat is doing just that. Exercising, exercising, exercising. And of course, reducing my intake. If I don't reduce my intake and I overcompensate by overeating, it's not going to make any difference. Overall, to lose weight, I've got to be burning more than I'm taking in. Make sense? Question?
Just one quick one. So when you're sick and you get a fever,
Yep.
are you using the same processes as while you're getting hot, or is it a different
Okay.
method?
When you're sick and you have a fever, what's happening in your body is basically the question. Okay? Your body's working overtime when you get a fever. You're making immune cells, you're fighting off an infection, and that involves additional metabolism, which is why the warm temperature increase happens. Jade?
So you said respiratory control is when you have tight coupling of
Yes. Tight coupling between so respiratory control exists when you have tight coupling between electron transport and oxidative phosphorylation, meaning you have an intact inner membrane, and when you have an intact inner membrane, one depends on the other. Electron transport won't run if oxidative phosphorylation is not running, and oxidative phosphorylation can't occur if electron transport is not running. Yes?
So in like the dynamics, when you talk about thermogenics, is this what it's affecting?
To be honest, I don't mess with thermogenics, so I will probably better not be one to answer that question. How about that? Okay. What I would say, most of the diet industry is baloney, so that's my basic principle there. Okay. I promised I would talk about electron transport inhibitors. Okay? So the movement of electrons from one complex to another can be inhibited by certain compounds. Okay? These two guys here, amytal and rotenone, can stop the movement of electrons out of complex 1. This guy right here is actually used as an insecticide. And it's actually, interestingly enough, considered an organic insecticide. It's actually produced by certain species of plants. And it's very effective on insects because insects are much more dependent and have a slightly different complex 1. Doesn't mean I want to go out and take a spoonful of it, but it's not as poisonous as the other ones that I'll talk about. Another reason that complex 1 inhibitors aren't quite as poisonous is because complex 2 is a way around complex 1, right? Now, I don't recommend it, but we can think of other ways of getting electrons into the system. Antimycin A is an inhibitor of complex 3. The last ones I want to talk about are down here. Cyanide, azide, and carbon monoxide all block movement of electrons through complex 4. These are the most poisonous ones of the ones there. You can't bypass them. If you have cyanide, you will stop everything. Imagine I stop here and I say, well, maybe I'll bypass it by getting something else in here. Or I block complex 3, which is right here, and I can bypass by getting something else in here. This is the last step in the process. I can't bypass it. Okay? Yes, Andrew? There are treatments people give if a person has cyanide poisoning, but they have to be given very early, and they're with very low efficiency. Okay? Carbon monoxide kills you 2 ways, folks. It kills you here, and it kills you by competing with oxygen for binding to heme. So it's squeezing oxygen out of the picture. All right? These guys are the most poisonous of all. Okay? Very nasty stuff. If I stop right here, what's going to happen to movement of electrons? It's going to all pile up right here, right? Any place I stop, electrons are going to start piling up, and I'm going to have problems arising from that. What if I gave cyanide, okay, to a cell? Tell me what's going to happen inside that cell. What's the first thing that's going to happen? What's the first thing that's going to stop? Nope. The first thing that's going to stop is going to be this thing, right? I'm going to stop transferring electrons here. That's the first thing that's going to stop. If I stop transferring electrons here, what's the next thing that's going to happen? These are going to pile up. And as these pile up, what's going to happen to the proton gradient? It will go down. There will be none. Because whatever there was is going to be used, and it's just going to fall off until it equalizes out, right? What's going to happen to my ATP concentration? It's going to go down. Okay? What's going to happen to citric acid cycle and glycolysis? They're going to stop because NADH is needed, right? Okay? This is why it kills me. Okay? My ATP concentration goes down. I have no way of making ATP because oxidative phosphorylation is completely hosed. So I've just given you some scenarios about how respiratory control works. I think these are fair game for me to ask you at some point. I want you to understand that.